Hello, everyone. Welcome. I'm Asiye Golabchi, your MC for today's webinar, and I'm very thrilled you could join us today. We got a fantastic presentation that will take us right to the cutting edge of electrophysiology. However, before that, I wanted to share our today's plan and also some housekeeping details. So we will kick things off by walking through the key tools featured in our speakers paper. I'll share a few other options that you might want to be aware of, and then we will hand things over to Dr. Duan Lee from University of Michigan for a deep dive scientific talk you probably won't want to miss. So for the housekeeping details, while we are rolling, drop your questions or your thoughts on the chat box whenever they pop up. I'll keep an eye on questions. I probably are going to answer some of them, and if they're not answered, we will wrap a live Q&A session after Dr. Lee presentation. All right, let's start. So one of the here, what you're looking at is the star of our show for today. We call it the SmartBox Pro. This is a data acquisition system that was used in the research paper of Dr. Lee. So think of it as a full-blown rack of electrophysiology hardware that's been shrunk down to the size of a paper bag, and price is just as under $5,000. So you get high-end muscle without a high-end bill, right? So maybe now the question is, what are the capabilities? So using this system, you can record up to 1024 simultaneous channels. There are plenty of headrooms for everything from a single shank to, let's say, a stadium-sized synapse arrays for our synapse user. They're very familiar with this system. So there are a flexibility of recording 1 to 30K samples per second per channels. That means this let you dial with any type of bandwidth that you need. So you're not wasting any storage on your computer. It has a built-in GPIO for closed-loop work. There are 2 analog and 2 digital TTL output for triggers or stimulation, plus matching input for behavioral or opto data. So there is an audio jack that you can listen for the spike when they fire up. So if you look at front of the box, you can see 4 HDMI ports, A through D. So you can plug in multiple probes at once. When you flip the box around, you'll find clearly labeled GPIOs. There is a USB 3 that connects directly to the computer for the fast transfer of your data, and the simple 9-volt DC battle for power. Regarding the filters and fidelity, there is an adjustable cutoff frequency from 0.1 hertz to 20 kilohertz. And using this frequency, you cover everything from a slow, let's say, DC shifts to high frequency multi-unit activity. So it has also the capability of detecting smallest signals of 15 microvolt. That means you can chase the low amplitude synaptic events with confidence. What are you looking here is our software, our custom software that we call it Radiance. So this software pulls the whole data framework together from live acquisition to deep dive analytics and offline. So this software controls a SmartBox Pro that I showed you in the previous slide. So Radiance bundles Lego, Curate, Vidiri into a single consistent interface. While you're recording, LEGO streams noise, SNR, raster plots, and spike sorting live to the screen, plus an interactive 3D digital twin of your probe inside the brain, so you can adjust the depths and the angle on the fly. Every dataset is served as an OpenXStat bundle with the rich JSON metadata, so it's easy to read in Python, MATLAB, or any other language. If you need automation, we have Python API packages that give you full programmatic control for closed loop experiment, or if even you need to do batch processing, it's possible using our system. Please keep in mind that the raw data, they stay local and traceable with unique IDs. So when you use radius, technically you turn the raw voltage that coming from your probe into actionable insight. You can have it in the live, offline, and everywhere in between by using the Q rate that let you manage your data. So to be honest, if you use Radiance, you're spending less time with the software and you have more time to focus on your experiment and the recording that you have. So if you have any question about the compatibility of this software, feel free to put them in the chat box and I will address them during the Q&A session. Okay, so now time is to zoom into the SmartLink head stage that comes with a SmartBox Pro. So consider if the SmartBox Pro is the brain of the system, these boards are the set of ears sitting right on the skull. So by digitizing the signal millimeter from the recording side, these head stages, they chop out cable-induced noise or motion artifacts that typically you're observing in free behaving recordings. So you can select the SmartLink head stages of 16, 32, and 64, and each of them are available in acute and chronic versions. So even for 64 channel chronic version, you can see the size is about 15 by 20 micrometers. So think of postage stamp size, right? So typically when you're getting your probe, your head stage and your SmartBox Pro, it has a plug and play workflow. So you snap your NeuroNexus probe into the SmartLink head stage, and then using one SmartLink cable that will carries the power, clocks, and digitized data straight to your SmartBox Pro. And then you fire up the software and you're recording. No pin mapping, no soldering, no headache. Very easy, very fast data collection. So as an alternative, we have other options for the users that they want to do electrical stimulation or our NeuroPixels users. We offer X-Stack family, and you can pick up the box that matches your science. So here is the bird eye view of our X-Stack lineup, 3 models. They share the same DNA, but a scale in muscle and GPIOs. So the entry point is Core 2 with the option of 512 recording and 16 or 32 channel Steam. The One Plus is the workhorse. It doubles the channel ceiling to 1024 and 128 Steam. And it drives 4 NeuroPixel probes in one box. It has the same HDMI ports, just more lanes on the highway, right? Compared to the Core 2. So all in one is the flagship. It takes the One Plus guts and folds the 4 LED drivers for optogenetics, plus 4-channel HD video capture. That's again perfect for closed loop behavioral studies if you're interested. So again, whether you're piloting a 64-channel rodent study or planning a 1,000-channel multimodal primate recording, there is always data acquisition system that we can offer you. And you can switch the models. Either you go with the SmartBox Pro if it's recording only, or you can select our let's say X-Stack family. Again, if you have question about this system, feel free to ask and put your questions in the chat box. Here you're looking at the X-series head stage, tiny but mighty. So each little cube here is about the size of your thumbnail. You can look at this picture and check the penny in the photo for the scale. So it has everything that you need to digitize the neural signal right at the implantation site to keep the noise to the minimum. You can pick from 32 channel or 64 channel models in both record only and the version of record and steam model. Or if you want to go big with the 128 channel, we have recording only version available. If you need to connect these header stages to your existing probe, probably you have noticed the connection part. Don't worry, we offer adapters that make this connection very easy. Again, if you have compatibility question, drop them in the chat box and I will answer them. What you're looking at this slide is the H64LP package probe that was used by Dr. Lee in her study. So here we are talking about 8-shank 64-channel probe. Each shank carries 8 recording sites that have 413 micrometer square area. They've arranged right along the edge for maximum tissue contact. The sites, recording sites, they're spaced 100 micrometer center to center, while the shanks themselves, they're 200 micrometer apart, again, center to center. So this design giving you a tidy grid for precise laminar mapping. In terms of geometry, every shank is 2 millimeter long, but only it has a 700 micrometer active area. So you can target a specific cortical or subcortical layer without picking up extra noise. Another thing, this whole array, it has a low profile package of X64LP package that comes with two 32-channel Omnetics connector. And using this flex cable, it attached to the PCB, let's say the probe attached to the PCB. So we offer different type of packages for this design. If you have any question, we would be happy to help you to match it with the header stage that you have. What you're looking at this slide is our SUMA framework or connectivity diagram that you can find on our website when you look for data acquisition system. Based on the whole solution that we offer, you're getting rack level performance, plug and play compatibility with every NeuroNexus probe and intuitive probe centric software suite that I explained to you earlier. If you need a steam recording or real-time closed loop, you can always set up the X-Stack family. But for most experiments, the SmartBox Pro gives you everything you need in one tidy, budget-friendly box. So if you have any question again, you can put them in the chat box. But I appreciate you guys for listening to my presentation. And again, I will answer questions during Dr. Lee presentation. So without any hesitation, I'm delighted to introduce Dr. Duan Lee. She's an associate research scientist at the University of Michigan Center for Consciousness Science in the Department of Anesthesiology. With a background in engineering and a specialization in neural signal analysis, her research focuses on brain dynamics, consciousness, and anesthesia. A central aim of her work is to develop advanced electrophysiological signal analysis methods to investigate the neural mechanisms underlying altered states of consciousness, including anesthesia, sleep, and psychedelic states. Dr. Lee, the screen is yours.
Hello, everyone. Thank you, Ozzy, for the candid interaction and also for the opportunity to provide us to present here. I'll start my sharing. Okay, here's my screen and then my pointer.
Yes.
Okay, so let us start. So today I'll present our findings on how anesthesia modulates the complexity of neuronal firing in the rat neural cortex. Before I present our work, I'll start just from a brief background. While natural sleep has well-documented sleep transitions across multi-stages of sleep, general anesthesia has long been assumed to produce stable dose-dependent brain activity. For example, here, as anesthesia deepens, EEG signals are thought to switch from more awake-like pattern to burst suppression in a dose-dependent manner. However, recent studies found even at stable anesthesia, multi-brain states can occur. In this rat local field potential study, the middle panel shows the time-resolved spectrogram from multiple brain regions during stepwise decrease in isoflurane. So we can see abrupt global changes in spectral power, and these changes don't follow the changes in anesthetics. Clustering these spectral features reveal the multi-state can coexist at the same concentration level, and the brain can frequently switch between them. Clinically, this is concerning because the occurrence of a more awake-like state at deep anesthesia, for example, here, the state in right, maybe increase the possibility of transient awareness. Although this evidence in local field potential, their underlying neuronal basis remained unclear. So to fill the gap, our lab designed a study to investigate the state transitions in cortical unit activity. We used the 8 rats. Each rat implanted with a multi-electrode array in the right visual cortex here. We targeted deep layers 5 to 6. For anesthetics, we used the desflurane, which reduced in a stepwise manner from 6%, 4%, 2%, and 0, no desflurane. At each level, we waited 15 minutes for equilibration and then recorded 20 minutes spontaneous activity, followed by 5 minutes visual stimulation. The stimulation was emitted by a red LED, which was affixed to the cranium behind the left eye. So the flashes had a duration of 10 milliseconds, repeated 100 times at each level. The interstimulus interval are randomly from 2 to 4 seconds. So we concatenated the recorded signals across these spontaneous and stimulus sessions and do the pre-processing. We first filtered out the spike and the local field potential. To reduce motion activity, we median reference the data and divided them into small windows, 1 second windows. If there are any noise detected in any window, we just set them to 0. After pre-processing, we use spiking circuits for spike detection and classification, and then followed by manual curation in Phi. We excluded 2 experiments with low number of sorted neurons. Finally, we have 6 experiments for the analysis. Here, we showed an example from a representative experiment by concatenating the different anesthetic levels. We see sorted spike trains, 1 trace of local field potential and the corresponding power spectrogram. At the same concentration level, we see there's different patterns, not only in local field potential, but also in spike trains. Our aim was to identify the different states in spike trains and investigate how these different states modulate the cortical bearing. To achieve this aim, we started from spontaneous signals concatenated across different anesthetic levels. We use principal component analysis of PCA to get the population activity, then extract features and use the clustering to identify different states we call population states. Then we examine pre-stimulus data to examine whether these spontaneous states also appear during pre-stimulus. After this population state defined, we investigate the state-dependent spontaneous bearing and also the stimulus-related responses at 3 levels. First, we look at individual unit bearing, and then we look at neuronal coactivation, finally neuronal complexity. Let's first look at how we classify the population states. We started from the spike trains. We binned, smoothed, and Z-scored the spike trains, and then applied the principal component analysis. We retained the first 2 components. This is determined by a comparison with surrogate thresholds in terms of explained variability in explained variance. So for each component, we extract features. The feature we used just simply the mean activity and the temporal variability, quantified by permutation entropy, or PE here. So in the feature space, we applied a density-based clustering to identify different states. In this experiment, it shows 5 clusters. 4 of them showed progressive decrease in both mean activity and permutation entropy. The last one deviates from this pattern. On this top plot, we show the state time sequence. So at a constant anesthetic level, we can see multi-states can coexist. For a single state, for example, this state in orange, which can appear at a different anesthetic level. This means the cortical unit activity are not stable. They are dynamic. They are not correspond one-to-one to those. Of particular interest of this state in blue is mainly occurred at deep anesthesia, but unexpectedly it showed high PE variability. So we call this state paradoxical state. Across all experiments, here the first panel showed occurrence rate of each population state at different dose. And the second panel showed the state-dependent population features. We see from state 1 to 4, both PC1 mean activity or spike rate and also the permutation entropy variability gradually decreased. These 4 states mainly reflect the deepening anesthetic level, but not specific for a certain level. State 5 mainly occurred at deep anesthesia, but showed high PE, comparable to that in state 1, the weak state. As for the spike rate, it's also elevated from state 4, but not as high as the PC1. To relate this, spike-dependent state to more familiar local field potential. We can see here the state 1 mainly. Okay, so state 1 is mainly in wake state, featured by high theta power. State 2 and 3 showed gradually increased delta power, consistent with sedation state. And state 4 showed the burst suppression. You can see here the high suppression level, 50%. State 5 showed the unexpectedly low amplitude in local field potential and without burst suppression. So with the spontaneous population state defined, we now look at the pre-stimulus period. Here is pre-stimulus data from all trials, from 1 second data from all trials. We concatenate them and apply the same principal component analysis and calculate the mean activity and the permutation entropy. In the feature space here, the shaded area represents the 80% confidence region of spontaneous states. This was generated by fitting a Gaussian mixture model. We ascend each trial of pre-stimulus state to the nearest spontaneous state. After that, we found the distribution of pre-stimulus data are largely overlapped with spontaneous ones, but just the distribution a little bit wider. This is probably because the effect of repetitive stimulation. To quantify this mismatch, the bottom panel shows the city block distance between the spontaneous state center and the pre-stimulus state center. On the diagonal line, these values in dark color means low distance between the state centers, suggests a strong correspondence. Again, the top panel here shows the state sequence. This suggests the pre-stimulus data are also dynamic. It's not to mention, besides the 5 states we defined previously, and we defined another state called the suppression state. In this state, we include all the trials where all the neurons were silent during pre-stimulus 500 milliseconds. This definition was intended to differentiate the trials with inactive baseline. We see this state mainly at deep anesthesia. So across all experiments, the pre-stimulus states largely overlap with spontaneous ones. We also see here from state 1 to state 4, the gradual decrease in mean activity and the permutation entropy, while state 5 showed a high PE comparable to state 1. It's as expected, the suppression state shows the lowest mean activity and the lowest permutation entropy. So far we have the population state defined in both spontaneous and the pre-stimulus activity. We then look at how these states affect single neuron firing. First, we classify the neurons into narrow spiking and wide spiking as putative inhibitory and excitatory neurons based on their waveforms. specifically the trough to peak duration. Across the states with deepening anesthesia, we could say both steps showed a significantly decrease in spike rate. But if we pay attention to the state 5 in the wide spiking neurons, the blue color, it's elevated from state 4. We then look at the autocorrelogram or ECG across states. We still pay attention to the wide spiking neurons, so the distribution became more peculiar with deepening anesthesia. This suggests more bursty activity. This effect was quantified by ECG peak time and the ECG peak ratio. The ratio is between this peak value and the baseline. Well, in state 5, this particular distribution was not very pronounced. We also look at the interspike interval or ISI. First, from a few example neurons. Here we show the ISI raster plots and their frequent distribution, and also the distribution across all neurons. We still look at the wide spiking neuron in blue. So in baseline stage 1, so there's a single peak. But with deepening anesthesia, there's multi-peaks. And then this multi-peak return to 2 peaks in stage 5. This observation was confirmed by the modularity analysis of individual neurons, as shown here. So overall, our analysis suggests anesthesia reduced the firing and promoted multimodal bursty-like firing from stage 1 to stage 4. But this effect was partially reversed in stage 5. Based on these results, then we turn to the sensory responses. Here, we also show a few example neurons. Their post-stimulus raster plots and the average response across the trials, and also the responses at group level. So in stage 1, stimulation evoked a high peak here, then followed by a dip and diminishing oscillations until 1000 milliseconds. With deepening anesthesia, the later part of these responses were significantly reduced. But for the early part of the responses, the most significant change is the disrupted timing. We can see here the single peak here became split to 2. So in stage 5, the responses resemble stage 4, but the amplitudes were not surprised that much. And surprisingly, in suppression states, compared to other deep anesthesia states, the responses actually more stronger and prolonged for longer time. So far we have analyzed the individual firing. Then we look at neuronal synchrony. Here we use approach called the functional clustering algorithm, which was aimed to detect statistical significant ensembles from neurons. That is defined by a group of neurons that can spike together more often than expected by chance. This algorithm started from a significant matrix between every pair of spike trains. From here, for example, it first looked for the pairs with significant coactivation. Here is between S1 and S2, then merge them. And then in second group, find another pair, S3 and S4, and merge them until no merge was possible. Using this algorithm, we can identify the firing mode for each neuron. Here we show results from 2 example experiments. So for each neuron, it could participate example, maybe example 1 or example 2 here. We call these neurons as coactive neurons. Or maybe some neurons don't co-activate others. They just spike independently. We call them independent neurons. Or maybe they still spike, but not very fast. Their mean spike rate lower than 1 Hz. We call that inactive neuron. From here, we can see the spiking pattern. Oh, I forgot to mention, each dot here means 1 neuron. So we can see the neuron may, across states, they may remain in the same state, in the same spiking pattern, but they may also switch between different modes. So with this approach, we first analyze the spontaneous states. The first panel, we show the fraction of active neurons. We could say from state 1 to state 4, there lies the number of active neurons become less. This is consistent with the reduced firing rate as we described before. Well, of these active neurons, when we calculate the fraction of coactive neurons, the fraction increased. And also, we quantify the coactivation rate. That means how often the coactive neuron spike together with other neurons. We also see the coactivation rate increase from state 1 to state 4. That means the deeper anesthesia states demonstrate more synchronized activity. In contrast, in state 5, we go back to here. So there's, compared to state 4, there's more active neurons. But of these active neurons, less of them co-activate, and the co-activation rate was low, comparable to that in state 1. That suggests the state 5 is an asynchronous state. Then let's look at with stimulation. Here I showed the same 3 panels. Look at the fraction of active neurons, coactive neurons, and their coactivation rate. But for pre-stimulus 1 second data and post-stimulus 1 second data separately. As in spontaneous ones, we also see coactivation rate increase from state 1 to 4, suggests synchronized. In state 4 and also in suppression state. But in state 5, still is low coactivation rate, suggests asynchronous. So far, we have examined the single unit firing and also the neuronal coactivation. State 5 behave different with other states such as state 4, suggests this state is a paradoxical state. Then finally, finally, let's look at neuronal complexity. We start from spontaneous data. To quantify this, we binned the spectrums and divided them into 1 second window. So for each rat, the y-axis here shows the neurons and axis is time. Here, this black dot suggests a spike in that certain bins. To quantify the complexity of this pattern, we use lamp-wave complexity, or LZC. LZC measures how diversity of distinct patterns in this matrix, but it's sensitive to the changes in the firing rate across the states. So we normalize LZC by spike entropy. We got the ICI index, is spontaneous complexity index. So ICI showed the significant decrease in all deep anesthesia states, including state 5. Let's extend this study to sensory responses. So again, here, the first row, we show a single neuron. This blue line shows their average responses across trials. We banalize these responses by baseline activity, got a binary sequence. For each rat, we did this for every neuron and get this binary matrix. In each matrix, we sorted the neurons based on their spike rate. So the right arrow here indicates the neuron I shown here. So let's look at these responses. In state 1, the responses are widespread across most of the neurons and across time with variable latencies. Compared to a deep anesthesia state 4, there's less number of neurons responded and mostly with shorter latencies. Suppression states, the responses are stronger and prolonged long time. So to quantify the complexity of these patterns, again, we use the LZC and its normalized version we call perturbational complexity index, or PCI. PCI was originally proposed in TMS EEG studies in humans. It measures how differentiated and integrated of these responses to external inputs. It has been found to be low in an unconscious state, including deep anesthesia and slow wave sleep, but kept high in wakefulness and REM sleep. We adapted PCI to our study. It showed here significantly decrease in all deep anesthesia states, including state 4, 5, and suppression. There's no significant difference between state 4 and state 5. So both spontaneous and stimulus-related complexity showed low complexity in state 5, suggests it's consistent with unconscious state. So to summarize, first we observed the multiple dynamic states at the constant anesthetic level in rat visual tests. These results challenge the conventional assumption of a single fixed brain condition at stable anesthesia. Second, one of the deep anesthesia states showed unexpectedly high asynchronous firing. So we considered it paradoxical, but its low complexity indicates at global level, it's more consistent to an unconscious state. Third, fresh stimulation during suppression evoked a stronger, prolonged responses. This suggests the role of baseline activity in shaping sensory processing. However, our study only used one anesthetic with recorded signal in local visual cortex. Further research will be needed to look at different brain regions or multi-regions and other anesthetic drugs and other species, or maybe using more complex stimuli. So for additional information, our raw data are available in database archive, and the related publications from our lab are listed here, including our code. Before I conclude, I thank my PI, Dr. Hudet, for his guidance throughout this project, and also for everyone in our lab for their support. Thank you for your time, and I'm happy to take any questions.
Very fantastic presentation, Dr. Lee. I really enjoyed. I'm sure our attendees also enjoyed your presentation. So I will get more time to our attendees, maybe as a first question, if you don't mind. I noticed you showed the data for the visual cortex. So how do you feel about the other regions of the brain, the regions that, for example, they're responsible for decision makings? How you feel the anesthesia is going to have impact on the stats of the brain for those regions?
In current study, we only look at the visual cortex because we are more interested about like how brain process sensory information and how anesthesia modulates this processing. Yeah, we have planned to look at like other regions. Yeah, but we haven't looked at that yet. Yeah.
Okay, perfect. One other question that I have, so did you have time to look at the specific cell type and the effect of anesthesia on them? When I say a specific cell, like did you look at the response of excitatory or inhibitory neurons to the effect of anesthesia?
Oh yeah, it's a good question. Yeah, we have looked at like we have divided the neurons into narrow specking and wide specking neurons as putative like inhibitory and excitatory neurons. So generally, in terms of the failure rate, both type of neurons significantly decreased with deepening anesthesia, except the paradoxical state 5 here. So, and also we look at the ACG, the autocorrelogram. The changes in ECG pattern are more obvious in wide spectrum neurons. That means putative excitatory neurons. It suggests there's more bursty spiking with anesthesia. And also in ISI distribution, interspike interval distribution, also I think our results suggest wider spike neurons. The excitatory neurons are more affected by anesthetics than inhibitory neurons.
Okay, wonderful. And I'm asking my last question and then let the audience to also ask their question. So for the future, do you think there is any potential for using stimulation to change the state of the brain. For example, if you want to awake the person from unconsciousness to the awake situation, do you think it's gonna something with the very small stimulation could happen?
Yeah, I think it would be a fantastic direction to explore in future. I think the targeted manipulation of certain neurons or certain circuits will be very helpful for us to identify the role of these neurons and these circuits in state transitions and anesthesia.
Okay, perfect. So we have a question. So Manish, I apologize, you raised your hand after my presentation and I didn't see that. So, but now he has a question. Why in LGS continuous synchronous firing occurs? If you could answer that, Dr. Lee.
LGI. Yeah, I'm reading this question. I'm not quite sure what means LGI. Continuous.
I think it's a place in thalamus.
Salem.
So Manish, if you don't mind, I can let you to speak. Is it okay? Manish, you can talk if you want.
Yes, yes. Yeah, you can talk.
No, if you want to ask your question, I think Dr. Lee need more input from you.
Yeah. Actually, my question is that in Linux cluster syndrome, some neurons firing continuously occurs. What is the cause behind this? Do you follow my point?
I apologize. I think I didn't quite follow like what means like LGIs.
Actually, the question is related with seizure, continuous seizure.
Seizure, continuous seizure.
Yeah, yes. That is
Continuous
epilepsy.
epilepsy.
Yes.
Did you mean the paradoxical state I define in this study? We see the continuous spiking in deep anesthesia, but not just suppression.
No, not related with anesthesia, but in normal condition, interictal EEG shows that continuous synchronized firing occurs. What is the reason behind this? Not in deep anesthesia.
Okay.
Do you follow my point?
I think
It is the interictal EEG pattern. Yes.
I did. Yeah, if you don't mind, maybe we can follow up after the session. Yeah, I didn't quite follow your question here. Yeah.
Okay. Thank you.
No worries. Thanks, Manish. I appreciate you. Do you have any question about the part that I presented?
No.
Okay, perfect. Thank you. So we have a question from Elena Semernia. I apologize, Elena, if I didn't pronounce your name correctly. What light spectrum was used for LED stimulation? She's asking for the wavelengths.
I'm not quite sure the wavelength, but we use the red light LED. Yeah. We affix that to the cranium behind the left eye.
Okay, we have a question from Tom. Hi, Tom. What anesthetic or anesthesia state would you recommend for maintaining the network complexity of waking or sleep states? Or would isoflurane, desoflurane, urethane, ketamine, etc., all be capable of producing similar stimuli responses or network states and instead be a question of dosage?
Let me first address the first question. If about what anesthetic or anesthesia states would you recommend to maintain the network complexity of waking or sleep state? We haven't tested other anesthetics in this study, but based on my observation in other study using humans, I think I would expect ketamine would show ketamine would show high complexity because it might suggest the internal consciousness with ketamine, especially the low dose ketamine. For the sleep state, I think most of the GABA type anesthetics induce some states has some similarity with sleep, but not same. In human studies, I would think dexmedetomidine anesthesia may be more closer, have more similarity with sleep state. Yeah, for the second question, would isoflurane, desflurane, and all other ketamine capable of producing similar stimulus responses? We haven't tested in this study, but as I recall, in one of the previous studies from our lab, they tested urethane anesthesia and isoflurane, desflurane. I think mostly the pattern would be similar, but not same. But I think we haven't tested the stimulus responses using different anesthetics yet. Definitely would more study in future.
Okay, perfect. And then we have question from Irene DeMarco. You use flashes as a stimulus. Do you think that you would obtain different results, more or less reduction of activity due to anesthesia with more complex stimulus such as gratings?
Yeah, I think it's possible. It's possible. Yeah, our current study, we just use the light flash. But we use affix the light to the cranium behind the eye, so we can get like the similar intensity stimulation across levels and across different files. But in future, we definitely need to look at like more complex stimuli, or maybe even try some cognitive tests to better understand the phase transitions during anesthesia.
Okay, perfect. I don't see any more questions. I appreciate again, Dr. Lee, for your wonderful presentation. And I appreciate everyone for being with us and attending this webinar. We have recorded this meeting and we will post it soon, so please stay tuned. And for the future webinars, if you're interested to present your research, please feel free to reach out to me directly, and I would love to have you one of our speakers. Without any hesitation, take care, everyone, and I close the meeting. Thank you. Bye-bye.
Thank you.